All right, good morning, everybody. Feels odd starting on a Wednesday, but I kind of like it. All right, so we are jumping ahead a bit. We're skipping over the microbial genetics chunk of material and moving into thinking about how we deal with the microbes from multiple ideas with this. This chapter, we want to think about mechanisms that we might use to deal with the growth, like maybe in a lab situation, Bunsen burners or something like that to sterilize stuff. Talk a little bit about autoclaves and other chemical means to deal with microbes. You guys go into the lab and use the disinfectants to clean the benches before and after you do your stuff. Things like that we want to think about as we start moving into this chunk of material. After we do those initial kind of introductions with how we might kill organisms through chemicals, we will shift over and start thinking about stuff like antibiotics and then the actual immune response and how our levels of immunity help to combat these foreign invaders. And we'll also talk about how that starts to break down and how organisms have the potential to somehow skate around our immune response, or potentially for our immune response to go haywire. And we'll think a little bit about some of the autoimmune diseases that can result with some of that as well. That's kind of the whole chunk of material in this unit. And we will spend some time thinking some details of that as we get going. So for the control of growth, there are all sorts of things that we might think about, and we try in the lab to show you these things and let you think about aseptic technique and trying to be as clean as possible as we're going about this process. And really, that's kind of what the focus of this chapter is all about as we're trying to control this growth from those standpoints. I like this image. It's not the same as your first lab, but it's like that. You had the opportunity to go around some stuff. I think we kept it within the building. However, looking at this, it kind of makes me wonder, what sorts of things could you have potentially found if you went and swabbed your car? Maybe you don't want to know that, right? You probably don't. I have done a couple of outreach things where I've gone into some schools. Oftentimes it was with my kids, and I'd go into their school and we would do like hand washing experiments. We'd swab our hands and see what sorts of things were growing. And teachers liked to swab some other stuff, checking out what things could be growing in their classrooms. And occasionally a cell phone gets busted out and they swab that as well. And that's always pretty disturbing, all right? Things that you come in contact with that you're touching frequently, you would expect that you would find more organisms possibly in those situations than something that's receiving less contact. And that's really kind of what this is showing here as we look at some of these details as far as how many things we might be seeing. We're not saying what we found, but we found stuff, all right? All sorts of different types of microbes. And the CFUs up at the top, number of CFUs, this would be plating out these organisms and individual colonies that start to grow on those plates. That's a CFU, or a colony forming unit. The more of those you have, the greater amount of contamination we might think about, all right? So there are numerous touch points as we look at this, with definitely more opportunities for things to be possibly growing. The center console through the roof with things that are in there. And there's the possibility of all sorts of things getting touched, but also maybe some other stuff maybe getting spilled. Thing that sets the stage to providing some good nutrients for that might lend increased opportunities to find extra things. So I kind of like this, and I'd like to sort of see some of those colonies just to see the variation. I'm sure when you looked at those environmental plates, you saw all sorts of weird things. And I know there were some where you could see some mold growing on those plates. There are organisms all over the place. And just simply leaving the lid off of a Petri dish, you'd find organisms growing. You don't have to go swab stuff and put it on there. Just in the air, there are organisms that in turn, given the proper opportunity, could land onto that plate and start to grow over the course of time. It's kind of interesting stuff. So we've shown some of this before. We've looked at a little bit with like the different safety levels of this BSL-4, where you've got the spacesuit on, if you will, that's negative pressure, so it's all blown up with air. The hood itself is a negative pressure, so it's being sucked into the hood so that whatever you're working with doesn't contaminate the lab. And in turn, your suit helps to protect you as well from whatever you might be working with. Definitely a mechanism that, and you can see, we saw before, the sleeves taped to the gloves. It's a similar idea right here. This looks like some heavy duty tape, putting it on them. So we look at this with the different safety levels, and we've mentioned briefly, like those spacesuits at the top of our list, the BSL-4 or biosafety level 4. This is like extreme precautions, warranting special laboratories that keep everything in there, so you would hope, and suits that keep you protected as well, all right? And we think of working with, some serious pathogens, maybe something like Ebola or other hemorrhagic fever organisms. Marburg is another one like that. Both opportunities to set the stage for some serious issues or outbreak situations if they weren't maintained in a controlled environment, kept isolated, and the individuals working with them protected, right? As we I can't with my phone. As we move down, the biosafety level 3, these are pathogens that, well, in a teaching situation, we don't work with these things. These are potentially indigenous or some exotic species that have the opportunity for some serious diseases. Not that the top one doesn't have those same issues. It's definitely more of a concern up there. Definitely issues with this, and it is likely something that we would think of as being transmitted through the respiratory tract. So droplet transmission or something like that could lend to those issues. Not necessarily thinking spacesuit situation, but definitely working in the hood with the negative pressure, keeping everything within there is definitely something that would be happening. You would be wearing some protective clothing as well, but not necessarily in the pressurized suits, all right? Face shields, other things like that might definitely be warranted, right? Stepping it down, this is where we start thinking more opportunities to come in contact with these, maybe just naturally, and it's also organisms that we definitely think in a lab situation, all right? So indigenous, we think of sorts of different levels of disease that they may cause and all sorts of mechanisms for transmission, more than just respiratory. Staph aureus, a common skin organism, also sets the stage as being a pathogen if given the right opportunity. And We also see some serious drug resistance with some of these guys, and it sets the stage for some big concerns, all right? So there is definitely caution, and I feel like any level of organism or level of biosafety that we're looking at, you should treat everything as if it is a potential deadly pathogen. That ensures that you will be safe when you are working with these. If you treat them as if they could kill you, I think that's a better way to go into it, all right? Probably not something that is super high on the radar. This is gonna, kill you, but it could. As we bump it down to biosafety level 1, this is maybe a little or where we would like to be in our teaching lab situations, keeping things at a minimal level of risk, all right? So these typically aren't known to cause disease in healthy hosts, minimal risk. So non-pathogenic strains of E. coli as an example, all right? With that said, I feel like, exposure to E. coli, bringing it into your body, whether we're talking pathogenic or not, you're probably going to have some ill effects with that, all right? But not something that's going to truly make you feel like you're dying, all right? So most of the organisms that we're working with in the teaching labs fall within this BSL-1 category. There are a few that we step it up and think of as BSL level 2, stuff like Staph aureus could fit within there. Shigella that can cause some pretty nasty diarrhea could fit in a BSL level 2 as well. There are a few microbes that we would classify as pathogens or potential pathogens, all right? With that said, the organisms that do fit in that category that get brought into the teaching labs, really all of them, even in this BSL level 1, are attenuated. And what we mean by that is they have been cultivated repeatedly. They have been manipulated through these processes of growing them that basically makes them weaker. It makes them lose some of their properties that could initiate a disease process in a person, making them safer to work with, all right? With that said, it also may set the stage to some issues when you're utilizing some specific tests to try to identify them, and maybe through the process of attenuation, they've lost some of those characteristics. It may make it a little harder to use those tests to truly identify. But most of the things we're working with, we've got stuff that fits and works just fine with them. So definitely different situations and different laboratories you may see yourself in a situation where you're working with things that might fit within this BSL level 3 category, tuberculosis, or some other highly contagious pathogens could fit within that category. Pretty unlikely that you're going to find yourself in a BSL level 4 situation. You could, you don't know, right? But usually these we're not thinking, usually thinking a hospital or a clinical sort of situation. It's more research oriented when we're thinking about that, trying to deal with it. And definitely, as we see here with the CDC classifications, Center for Disease Control definitely has laboratories that have level 4 capabilities. So too do the National Institutes of Health, where they are doing research with organisms that are serious pathogens, all right? So it depends on the circumstances and what you might be doing to begin with. So we've got a handful of different potentials that we might employ for controlling growth. And in some circumstances, we might be minimizing or inhibiting and not necessarily killing. And we can flip it around and say, well, we want to completely remove anything, all right? We also have it broken up into the fomites, the inanimate objects versus living tissue. Stuff that you would use on the bench top is highly likely not something you would be using on your body, all right? There would be some issues, probably some serious irritation with that if you were trying to put that on there and trying to cleanse with it, all right? So we'll start with the fomites, again, the inanimate objects, just things that you may come in contact with, touching. All of the things in the car that we just showed with all of the colony forming units, you could classify a cell phone or the mouse or the remote, all of those fomites and things that are touched by more people set the stage for all sorts of transient organisms, potential pathogens to be deposited onto those things and then picked up by somebody else, all right? That's a potential issue, all right? So with the fomites, we've got three different things. We can think of disinfection, sanitization, or sterilization, all right? And it definitely depends on what you're working with and what you potentially could have as a possible culprit that may set the stage for needing a different thing that we use, all right? So we'll start off. Disinfection reduces or destroys the microbial load of some inanimate object. Heat or antimicrobial chemicals are used as disinfecting agents. Your bench disinfectant fits in that category. It's called cytochrome. So the benches, clinical surfaces, bathrooms, all of these, usually the disinfectants are highly effective, all right? Chlorine, bleach, phenols, glutaraldehyde, all of these are potentials. And cytochrome is a phenolic agent. Sanitization is a little bit different. It reduces the load to safe levels with heat or antimicrobial chemicals. We're not seeing destroy, we're reducing, all right? Disinfecting may not completely destroy everything, but you hope it does, right? Sanitization, we're not ever saying that it completely destroys stuff, but it helps to slow or minimize the growth. Commercial dishwashing with eating utensils have a cycle that sprays a disinfectant or a sanitizer, excuse me, on there. Public restrooms also potentially with some sanitizing. So detergents with phosphates finish in maybe your dishwasher, also with a bit of sanitizing properties. It's not just for hard water spots. So the sterilization, this is a complete elimination of vegetative cells, endospores, and viruses on whatever sort of inanimate object. We think about things like scalpels or surgical equipment, things that you would hope are completely devoid of any sort of organism. That's what you want. It says here, you know, needles used for injection, or you could go, you know, into a tattoo shop. You would hope to see a little autoclave and know that the tattoo needles have been autoclaved. And if it's some dude's basement, you better think twice about that, right? It's like, it's fine, right? I wiped it on my pants. It's clean, right? That's a potential issue, right? So steam like an autoclave, chemical agents and radiation all can eliminate all of those potentials, all right? I'm going to tell you a quick story because I think it's kind of interesting. It's also, it's a problem, right? So sanitizing and sterilizing are definitely different processes. And when we think about sanitizing something, we add some agent, we slow or inhibit the growth, but then given the proper opportunities, organisms that were still there and inhibited can start to grow again, all right? That's a potential issue, all right? But dependent upon the circumstances may not matter, all right? So in the production of alcoholic beverages, you need to put them in some sort of a bottle, some sort of a container, and in turn, you would hope that that container is sterilized so that nothing was in there. So when you put your stuff into that bottle, all you have is your stuff, all right? That makes sense, all right? Oftentimes, you can sterilize glassware by baking it in an oven for hours, all right? Usually 4 hours at 400 degrees Fahrenheit is a sufficient amount of time to basically say these things are sterilized, all right? It kind of begs the question, does that truly eliminate the endospores? Not 100% with that, but I feel like it should, all right? But I don't know, right? We're not, to kill a spore, we technically need to get above the boiling point. Dry heat may or may not actually do that, all right? So, as a microbiologist, I, at times, will produce some frothy beverages. I have a yard with some fruit trees, and in turn, I will turn those into wine. There was a year where our apple tree was completely packed with apples, and my thought was, holy cow, look at all of this. I must turn it into alcohol. What else would you do with all these apples, right? Eat them? So, as I am doing this and I am making all of this apple juice, my children and my wife both thought maybe some of this juice should stay juice so we could consume that and my kids could consume that. And some of it could go the other route and I could turn it into alcohol. Everybody's happy now, right? So massive amounts of apples, lots of juice happening. We also were making some dried apples and all of that stuff. That's beside the point, all right? So I decide I could just bottle up this apple juice. That would be fine. And I will bottle it in wine bottles. I'll just put corks in them and it will be just fine. These wine bottles, I did not bake in the oven. I had a sanitizing solution, an iodine solution that you can also utilize for this, but there are issues, all right? If those wine bottles had been sanitized and then they dry and then I fill them up with alcohol, it's going to be fine because the level of alcohol will be inhibitory to other things that potentially could have been there, all right? These wine bottles also possibly have some yeast, because that's what's making the alcohol in the first place, and sanitizing it should inhibit that, but then given the opportunity, maybe not, right? If all you do is put juice in there, you have given it lots of sugar, plenty of nutrients, and any of those yeast cells that were there and were inhibited, now they start to grow. And in some of these bottles, over the course of a few weeks, they started to ferment. One of the bottles blew a cork out, and I walked downstairs and there is a small puddle, a little flood. And there's no reason for there to be a flood. There's no source of water. And I can smell a sweet smell. Kind of odd. And then I look over and see these wine bottles and one with the cork laying on the ground. It has blown the cork out. My first thought is, it's an issue, right? Fermentation must have happened. There must have built up pressure to blow the cork out. I made alcohol, but I didn't want to make alcohol. I just wanted juice, right? So as I am evaluating this, I smell it. It smells okay. I must taste it just to see. It also tasted just fine. There was a hint of alcohol, but not bad. All right. I made a cider. All right. But by accident. All right. And it was the result of some of these bottles that still had some residual yeast. And all that happened was a sanitization, not a sterilization. And it lent to their growth over the course of, like I said, a few weeks. All right. Then it showed up. So it depends truly on what you're going for, of what you might be doing. All right. I could have used those bottles if I would have baked them, and that should have killed all of the yeast cells that were in there and not lent to them exploding, blowing the corks out. But all I did was the sanitizer. All right. Didn't know. All right. At the time, I just didn't register that that was going to happen. But as soon as it did, it all clicked and made sense. And I'm like, well, that's all I did was sanitize. I didn't kill those yeasts. I just slowed them down. All right. So kind of interesting. And then as my wife, as I bring up this fresh bottle that I could tell has started to ferment, and I opened it up and I'm like, I've turned it into a little bit of alcohol. She tells me that tastes better than the stuff you make on purpose. And I'm like, oh, that hurts. But it was good. All right. And I'm like, maybe we're onto something here. It's a natural fermentation. That's what they did back in the day. That's also a potential other issue. All right. Okay. Enough of that. Enough of the wine. So on living tissue, couple of different potentials. We don't have the same ideas that we do up above, all right? A little bit similar when we think about trying to eliminate these potential pathogens, but the way we go about it is definitely different. So antisepsis and degerming are the two techniques that we might utilize. Both of them reducing the microbial load. This first one, we think about some sort of antimicrobial chemical. The degerming, there is scrubbing and or the use of mild chemicals as well. So the first one, we think about cleansing the skin after an injury or before surgery. There's handfuls of things that might be utilized. Boric acid, isopropyl alcohol, peroxide, and iodine, betadine solution rubbed onto the body before an incision is going to be made. The degerming, we could think about hand washing, so soap or alcohol swabs, which also could potentially come up if we're thinking like a needle stick or something like that, alcohol before that. So we're not in neither of these do we see killing or the complete removal of that because you don't think about that on living tissue, right? It's definitely a different situation. So this image with the death curve, I feel like we should flip it back to the, a little bit that we skipped over, and we think about how microbes grow in the first place. Once an organism gets into its specific environment, it takes a little bit of time to kind of get a feel for these surroundings. And there's a little bit of a lag period, and then they grow exponentially. And then once, whatever situation they're in, once these nutrients start to deplete a little bit, once wastes start to accumulate, we hit a stationary phase. And it's a kind of a plateau where they're growing and dying at virtually the same rate. After a period of time, they start to die, and they die almost as quickly as that exponential growth, all right? That's just in a normal situation, like in a test tube or in a flask, something like that, all right? This death curve, as we're looking at this, we could think about it with them just growing in a flask, or we could flip it around and think about trying to evaluate these organisms with the application of some sort of substance, cleaning agent, or the sterilization of the autoclave, something like that. How quickly or how much agent needs to be applied, whatever it might be, could be evaluated to see this death curve, right? And we could make measurements over the course of time and basically plot that to show what's going on, all right? So we could see on this figure, we've got the logarithmic scale that gives you a nice diagonal line that's easier to extrapolate stuff, and then the arithmetic scale that is very hard to visualize what's happening in this first bit as we're dropping off very rapidly, all right? They're showing similar things. It just depends on the circumstances as far as how we're looking at it. Logarithmic scale is definitely easier as we're looking at this over here to go through and extrapolate the values rather than this, all right? And this, it's showing, you know, from time zero, the initial point, and we've got a massive amount of microbes, right? 10 billion at time zero. And then we get down here and we're looking at very low levels, right? We still have a lot of cells, but it's much less, right? And this, over the course of almost an hour is what we're looking at, all right? But initially we can see, if we're looking at this first bit, over the course of just 5 minutes or so, we've dramatically reduced this load, and then we bump it over another couple of minutes and we're very low, all right? Still almost a million, right? More than a million, 900 million. So this D value is the decimal time that it takes to kill off 90% of that population, or a 1 log decrease if we're looking at that, because it's all a factor of 10 as we're moving through there, all right? And again, all sorts of different potentials, all right? So the Bunsen burner right here with the inoculating loop, this is stuff you guys have already dealt with and seen in the lab. This guy is similar to the Bunsen burner, all right? It's just it's a different way of doing the same sort of thing, all right? So flaming the loop, very common with a Bunsen burner in the lab to kill off anything that was on there before you use it, and then after you've used it, to ensure that it is clean, all right? This incinerator, or bacteria incinerator, this incinerator takes it a step further. It's got a lot of heating coils. It's not turned on right now, but it's bright orange. Inside is a ceramic tube that gets super hot. The idea with this is to prevent the possibility of cells, when they're going into that Bunsen burner, there might be some things that get aerosolized as they hit that, and they kind of vaporize off of that and potentially set the stage for issues, all right? If we were working with the level 2 or level 3 for sure organisms, we'd use this incinerator. The loop would go in there, and as it's getting cooked, anything that's potentially aerosolizing stays inside of this chamber and it gets destroyed, all right? Rather than potentially being aerosolized in this flame. The way you put your loop in and deal with that can help to minimize that, but there is still a potential issue with that. And, you know, the level of organism on the loop can set the stage for those problems as well. So we saw before, I don't remember what chapter it was, but they were like heating up a slide on the top, on the outside surface of this incinerator, because there is a lot of heat coming out of there. You could use it for something like that. But likely you're going to use the Bunsen burner to pass your slide through there. And I can see there's a gas hose right here. There's a Bunsen burner hanging out over there. I'm pretty sure, all right? And this looks like we've taken a picture just right in the lab, you know, downstairs. There's all sorts of stuff all over the place. I'm like, geez, we're going to put this in a textbook. Maybe we could clean some stuff up a little bit. But then I'm like, it also looks like a science lab. So maybe that's really what they're going for. Pipette box and some lens paper, maybe some filter paper over here. There's all sorts of things that, you know, makes it look like a real lab situation. It is. So we talked about the steam, the autoclave or a pressure cooker for the complete destruction. When we think about sterilizing something, the autoclave helps us to get the temperature up above 100 degrees. We oftentimes think right around 120, 115 degrees Celsius, somewhere in there, and minutes, 15, 20 minutes of exposure to that to ensure that we have truly killed any sort of spore-forming organism and everything else that could be in there destroyed. So there are all sorts of opportunities for these things. They can be very small little benchtop jobs to gigantic. The autoclaves down in the basement of the biology building are large. You've got a cart that can go into it with numerous flasks, 8 or 10 flasks, like 2-liter flasks could be cooked at the same time in one of these autoclaves. There are others that, like maybe in a hospital situation, where the autoclave could be an entire room. And you would see 2 rooms, 2 sides to the autoclave. One room would be the dirty room where all of the things that need to be decontaminated go. And then it goes into the autoclave. It gets cooked for its specific amount of time. And then when that's done, you're not in that same dirty room and opening it up. Everything in there should be sterile. The other side is the sterile room. And that autoclave, when it opens up on the sterile side, everything in that room is sterile. And in turn, things get pulled out and you know they haven't been intermingled with the dirty stuff in the opposite side of the autoclave, all right? Ensuring that all of those scalpels and everything else stay sterile, all right? And this, I mean, it kind of looks like that's what we've got going on right there as he's programming stuff. But it could be, this is just the autoclave, but I'd like to say this big stainless steel thing over here is really the autoclave. There are all sorts of different ideas with things like this and applications. And in a clinical or hospital situation, the amount of things that are getting sterilized is tremendous. And they are basically being used all day long, all right, as we are running through that, all right, pretty crazy. So there are things that we can see, like in the autoclave, that we can use to see if the autoclave has done its job in the first place. There are special types of like masking tape. It's called autoclave tape. And this first tube has not gone through the autoclave. The tape doesn't have the black line on it. You can kind of see this lighter bit right here. This is the line. And if it hasn't turned black, it hasn't came up to that proper temperature, right? If the autoclave, if you think the autoclave is functioning and you pull your stuff out and the autoclave tape didn't change colors, it never got to 115 degrees, right? You know that it didn't function like it should, and then you can't trust any of your stuff as actually being sterile. You need to have the autoclave serviced, or you need to check your parameters that you typed into it, because maybe it never went to that proper temperature, right? And sometimes if an autoclave is used by all sorts of researchers, they may go in and manipulate stuff. There was one scientist years back, they were trying to extract DNA from bacteria, and there's all sorts of ways you can do it, but they were testing a technique where you autoclaved it, but you only autoclaved it for, I think it was 5 seconds. It takes about a minute for the autoclave to start dealing with stuff, and then it goes up and then it drops right back down. It probably takes 10 minutes total that it's in there. If you set it for 15 minutes at 115 degrees, that cycle really takes about an hour for it to come up to temperature, to reach the proper pressure, to do all of its stuff, and then drop it right down, all right? So they manipulated stuff, then we're in there trying to autoclave things properly, and the cycle only goes for a minute, and we didn't realize that stuff had been changed. You realize it when you hear it finishing its cycle after a minute, but not sufficient enough time to actually say those things are truly sterilized, all right? So you can test with things like this. There are other mechanisms that are maybe a little more sensitive. It's a little vial, and inside of the vial is a spore from Clostridium. Clostridium and Bacillus are two of our spore formers. And in this little vial, it gets autoclaved. If it is proper temperature, that spore will be killed. After the autoclave has finished its cycle, you can basically snap that little vial, and inside of it, there's liquid and where the spore should be, and they come in contact. If over the course of a few hours, it grows and changes colors, you know that it didn't get to the proper temperature. So things that are more sensitive, you might employ something like that instead of this tape. Those are definitely a little more time consuming to ensure that it's happened. So the thermal death point of a microbe is the lowest temperature where all of the microbes are killed in a 10-minute time window, all right? That's the death point. The death time is the total length of time needed to kill all of the microbes at a given temperature, all right? It looks like it's about the same thing, all right? But this, here we've got a specific amount of time and the lowest temperature. This is not a specific amount of time, all right? It's how long it takes overall, all right? So they're similar, but not exactly the same idea. Lowest temperature and greatest amount of time. And the temperature in there is going to be varied. Shifting over a little bit, there are all sorts of other ways that we could think about possibly dealing with substances to ensure that they are safe. And pasteurization is a mechanism that kills pathogens, but it doesn't necessarily kill everything, all right? Pasteurization, and it depends on the circumstances, but we could think about regular pasteurization, bringing something like milk, which is really the first stuff we start thinking about with pasteurization, bringing it up to about a steam, not to a boil, about a steam, 72 degrees Celsius. And we hold it there for a few seconds, and then you're good to go, all right? Anything that is a potential pathogen can't tolerate temperatures like that, but there are other microbes that are thermophiles, things that can tolerate higher temperatures. They may not be targeted by this pasteurization. They're not going to make you sick, but we haven't completely removed all of the organisms, all right? There are still some potential things in there, but again, not something that's going to make you sick, all right? It's the others like the, Campylobacter, Listeria, E. coli, whatever, all right? Pasteurization helps to target these, but those that might be more heat tolerant aren't removed by that process. As we shift over to this ultra high temperature pasteurization, we bring it above the boiling point, and it's a very rapid thing, all right? So up to 138, it's held there for 2 seconds or more, just brief heating and then cooling. At that point, this stuff, and you can go to a supermarket and see some of this Horizon milk or whatever this one is right here on the shelf, right? Not in the refrigerator section. This is able to be sealed in an airtight container and kept without refrigeration for a handful of months, right? And all fine and good, right? As soon as you open it, that's a different story, and it needs to be refrigerated, all right? But until then, it's safe. And at this point, at this temperature, those thermophilic bacteria that are still present in this type of milk are wiped out over here. We don't have other organisms. I am not a milk consumer, but I can tell you that the flavor of these are slightly different because of the way that the milk is being pasteurized. And it alters some of the sugars within here, making the ultra pasteurized a touch sweeter, apparently, right? I personally think, unless it's a block of cheese or a tub of ice cream, I don't want to think about it in these containers because I don't want to drink it, right? But I have heard that it's different. Anybody know that? Anybody tuning in from home? You got some of this milk in your cupboard? There are other potentials where it might be irradiated. More common in European nations, but there are some potentials to see some of this. And it's a similar idea. This milk can be out on the shelf, not refrigerated, and technically longer than 90 days. I'm sure that it's able to be there. And to me, that just seems kind of gross, that it's able to withstand that amount of time. Like, I don't think that milk should still be good, but it is, all right? Kind of crazy, all right? Okay, I think that's a good stopping point. I'm sure you're all probably thirsty for a big glass of milk now. So that's what you should do. Go test it out, see what you think. All right, have a good day. We'll pick up with more of this Friday.